Bismillahirrahmanirrahim and welcome. In this topic, we are going to discuss the remaining topic of biomolecules. That were the protein, RNAs, and conjugated molecules. Let's discuss what are the proteins. Proteins, we have already known that protein are the most important and most abundant organic compound to be found in living organisms. They are composed of 50% of the derivatives, excluding water. They are present in all type of cells and in all parts of the cells. Every enzyme or the proteins, every hormone, almost most of the hormone are proteins. There are different functions performed by the proteins. We will discuss about different function of the proteins. They build the structure of the cells. Enzymes are the proteins, hormone of the proteins, and regulate the metabolic processes. Some protein like hemoglobin work as carrier and transport specific substances like oxygen, lipid, and metal ions, etc. Some protein are antibodies. They defend the body against foreign invaders. Blood clotting protein help in blood clotting. The movement of the organ and organisms during the movement is also by proteins. There are a lot of the function performed by proteins: regulatory functions, enzymatic functions, catalytic functions, and carrier transports, ion channel transport, intracellular transport, extracellular transport. There are various type of the proteins that are involved in different kind of the chemical reaction of the body. Proteins, now we will discuss about composition of proteins. They are composed of amino acids. When the more amino acids combine together, like 3000 or more amino acids are combined together, then they are called proteins. Amino acids are composed of nitrogen, oxygen, hydrogens. We will study their structures. About 170 amino acids have been found in the tissues and cells, but protein are composed of 25 amino acids, and most are composed of only 20 amino acids. These three are the most important MCQs with reference to MDCAT. And then we will discuss about general structures. All amino acids contain amino group and carboxyl group and variable R group and and a hydrogen atom attached to same carbon atom like alpha carbon atoms. This is the basic structure of the amino acids, and how amino acids combine to form proteins by a dipeptide bond, peptide bond, in which the amino group of one amino acid and carboxyl group of second amino acid react together, and one water molecule is excreted and dipeptide form, then tripeptide. Like here is the glycines, here it's carboxylic group, and alanine, here it's nitrogenous groups, NH2 groups, combine to forms a peptide linkage CN bond. Here is the peptide bond and the formation of the water molecules which removed from the glycines, carboxylic group, and hydrogen removed by amino groups, and resulting compound form is a dipeptide, consists of two amino acids and one peptide bond. It is a dipeptide called glycyl alanine. In this way the dipeptide are formed. Then we will discuss about structure of the proteins. There are various type of structure of protein, like primary structure. Primary structure is actually the sequence of amino acids in a chain. Number and sequence of amino acids is most important for the primary structures. It was first discovered by F. Sangers, who discovered the structure of insulin molecule composed of the two chains, alpha chains and beta chains. Alpha chain, total amino acids were 51. Alpha contains 21 amino acids, and other beta chains contain 30 amino acids. And in the same way, after quaternary structure of the hemoglobin were produced, two alpha and two beta chains. Alpha chain contain 141 amino acids and beta chain contain 146 amino acids, and total amino acids 574 amino acids. These are the important MCQs that are with respect to MCQs. Then there are different type of proteins, like almost 10,000 protein are formed. They are formed by the arrangement of 20 type of amino acid, like different words of English language are formed by only 24 alphabets. Same like that, different type of protein are formed only by arrangement of 20 amino acids. As the arrangement of the alphabet wrong any particular word of English, in the same way the arrangement of disturbance in the arrangement of a particular type of amino acid will derange the structure of proteins. Likewise, hemoglobin is composed of 574 amino acids, and if only one amino acid in the beta chain is deformed, then it will converted into sickle cell anemia. Like glutamic acid, polar amino acid is converted into valine at position number 6 of amino acid. It is converted into the sickle cell hemoglobin, and it is unable to carry out its functions. Only one amino acid is changed in this, but this structure and function is both deteriorated. Then secondary structures, when there is the helic form, or there may be a beta pleated sheets, and they are stabilized by hydrogen bonds. It is called the secondary structure of amino acids. Like most of the fibrous proteins are in the form of secondary structures. Then alpha helix are mostly right-handed or maybe left-handed. There are 3.6 amino acids each turn of the helix. Then the tertiary structure of amino acids, in which the protein fold upon itself, forming a three-dimensional structures. They are called globular proteins. They are maintained by ionic bond, hydrogen bond, and disulfide bridges. These bonds are also very important. They are formed in near the water in the same way that hydrophobic are the buried inside, and hydrophilic amino acids are on the surface of the molecules. Like here is the hydrophilic amino acids. So then quaternary structure is formed by different type of the protein chains, polypeptide chains arranged together by hydrophobic bonds, hydrophobic interaction, ionic interactions, and hydrogen bond, not by the covalent bonds. And hemoglobin is a type of quaternary structures of proteins. Lie and here are the important structures, like sequence of amino acids is primary structures. Then bending is and coiling is called secondary structures. Then double coil is tertiary structure of amino acids. And more than two chains that are found in a protein, they are forming quaternary structures. Then classification of protein, there are two main classification: fibrous protein and globular protein. Their difference is very important with respect to the MCAT questionings. Like fibrous proteins, they are in the form of fibers. Their property, secondary structure is more important in them. Insoluble in water, they are non-crystalline, perform structural role, and they are example of silk from this silkworm spider web, myosin from muscles, fibrin of the blood clot, keratin of the nail and hairs. Then globular proteins, these are ellipsoidal due to the multiple folding of the polypeptide chains. Tertiary structure is more important than soluble in aqueous media, salt solution of acids and bases and alcohols. They can be crystallized, disorganized with change in the physical and physiological involvement. Example are enzymes, antibodies, hormones, etc. Here we will discuss about nucleic acids. The nucleic acid that is included in our syllabus is only the RNA ribonucleic acids, and we will study only ribonucleic acids. Ribonucleic acid, then we have to study the basic structures. Basic structure is composed of nucleic nitrogenous bases that are found in the RNA are cytosine, guanine, uracil, and adenine, and they are formed by DNA by special process called transcriptions. In the central dogma, like DNA is converted into RNA, and by translations, RNA converted into proteins. Then we will discuss about types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA it's is a messenger. It takes the genetic message from nucleus to the ribosomes, from where the particular type of protein are formed. They are special. Their length depend upon the particular type of amino acids. Like there are 1000 amino acids which have to be converted into protein. Then nucleotide sequence should be of three nucleotides. So for one amino acid, the three nucleotide of RNA are used particularly, and this percentage is a small percentage, only 3 to 4% of the RNA. Then we came across the transfer RNA molecule. These transfer the RNA molecule to the ribosomal site where protein is being synthesized. They contain anticodon for a particular type of codon of the messenger RNA, and when the codon and anticodon of messenger RNA are formed, then the amino acids that is carrying on the transfer RNA is removed and removed, and they are removed, and the chain that is forming from the transfer RNA is attached on other exit pole, is removed from that particular type of transfer RNA, converted into this particular type of amino acids, and in this way the protein chain is elongated, and the amino acids are transferring from the transfer RNA into the chain of the polypeptides. They consist of 10 to 20% of the cellular RNA, is small, only 75 to 90 nucleotides, and there are at least one amino acid for each amino acid of the protein. There should be a one particular type of transfer RNA. There should be at least 90 kind of transfer RNA. For human, there may be up to the 45 type of transfer RNAs in the molecules. They are here, like here is our messenger RNA. It contain here codons, and maybe it's consist of like for loops. Its structure is like such like that, and it contain here anticodons, and here it contain amino group, and it is transformed into the start chains. Then it converted into P, then it is converted into A, and when it is converted into A, its amino acid is transferred to the next amino acid, that is transfer RNA, that is occupying this page. So the transfer RNA that is at this point is empty, and at this point there are the two amino acids, and when this reaches to these points, it contains two amino acids, and when this particular type of transfer RNA reaches at this particular reaction site, then these two amino acids are broken down from this type of transfer RNA, and they are adjoined to the amino acids of this transfer RNA that is now at this point, and at this point there is the further and other type of transfer RNA. In this genes, there are the three amino acids in the chains, then there are four, then there are five, and in this way this chain elongated. At the stop codon there will be no further elongation of the chains, and the transcription is almost